last takers on a plate of food before we get started? Well, I'm Jesse Kleingardner, and I'm very happy to introduce our second departmental honors thesis defense of the semester. This one done by Noah Smith. So, a departmental honors requires an awful lot of passion, time, hard work in the lab. And so I want to tell you just a little bit of, well, historical ruminations, which I guess the older you get as a professor, the more you spend time doing. So this project was one that was actually launched by Noah. And I can remember the moment in which it was born, actually. We were together in biochemistry lab doing some project progress update when I thought, whoa, man, wouldn't it be cool if we could do some a directed evolution project and convert well, I won't go into the details, I'll let Noah explain the details of the project. And Noah was just immediately excited about the project of building something that nobody has done before, and already started planning the details of it for his work that he did over this past summer and over this past academic year in this first project that he's going to talk about. So I should say that Noah this is a departmental honors defense, and a defense means that he's going to describe his project, present publicly for you all, but then afterwards, the professors will stick around, we'll dismiss you all, and we'll ask Noah questions to probe his depth of knowledge on the subject. So that is part of the process of tonight. I should also say that, and then we'll make a final decision regarding departmental honors conferral. This isn't the only honor, though, that Noah was a candidate for. We actually gave him the Outstanding Senior Award in the our local American Chemical Society chapter. He received that on Wednesday night at our education night. And the prize for that was a very large Merck Index, which only us nerds really appreciate. But I know that his mother's been working on reading that whole thing in preparation. So I just want to say, before I let Noah take over, that I have immensely appreciated working with him in the laboratory. And as Anna pointed out during his Outstanding Senior Award on Wednesday, he has functioned much more like a colleague than a student, adding in his own creativity, an enormous amount of ingenuity and passion and effort to this project, and I hope you all enjoy learning about it. Take it away.
Hi everyone. So I want to say thank you all so much, first of all, for coming. It's such a nice day outside, and you guys are here with me on a Friday afternoon at 4. And so I just want to say that means the world to me. This project has been so much of my time, it's been so much of my heart, and I'm really looking forward to telling you all about the work that I've done over the past 9 months, or maybe closer to 12 months at this point. Just designing this system for directed evolution of the biosynthesis of Cytochrome C. And so, first I want to start you off with this Carl Sagan quote. Imagination will often carry us to worlds that never were, but without it, we don't know where. And this just is a really, really careful and articulate encapsulation of what it's like to do research. You step into this world of unknown, you start to do things that you've never done before, but more importantly, you start to do things that nobody has ever done before. And that's a really scary thing to do. It's a very difficult and time-consuming thing to do, and honestly, sometimes it's not fun. It can be frustrating, it can be consuming, all-consuming in your thoughts, but ultimately, it's just a really big privilege and honor to explore the world that God has created for us and to learn to do new things with it too. So Before we start talking about the science, I just want to give you a little bit of the background to why this sort of work is important. So I'm showing you here graphs of the global population over time, as well as our energy consumption and our carbon emissions. Now, these graphs are a little old, so that graph goes up to 7 billion, the world population of 7 billion, but we just hit 8 billion. And we're going to continue to grow. And so, as we grow, we're going to want to farm more land to feed more people. We're going to want to build more houses, and we need to heat those houses. And we're going to use more plastic because we like to use consumables for things. It's more convenient. We want to stay healthy, and so we take medicine. And those medicines are very unique and complicated molecules that take a lot of effort to produce. And in the process, we make a lot of waste. And so we're really stripping the environment of its resources. And we're maybe not being so careful with how we do that. Weather patterns are getting more irregular, and we're sort of tipping very precarious ecosystems out of balance. It's not good, and we need to do better. So we need to get creative with how we're going to move forward and how we're going to use our natural resources in order to serve the world and take care of the environment that we're supposed to be stewarding. So there are a lot of creative solutions to this, but one of my favorite is biological synthesis. What I'm showing you here is E. coli, and at its heart, it's really just like this little orchestra of complex molecular processes that happen all in parallel in a little packet. And all of those things come together to give you a cellular life. On the right, I'm showing you the central metabolism of E. coli, and that's all of this area in white. But all of the colored boxes are actually engineered pathways that work in parallel to the metabolism of E. coli to produce natural products or similar compounds. So you might recognize a few. Down here, there's hydrocodone, which is a common painkiller. There's quercetin, which is an antioxidant. There's also erythromycin, which is an antibiotic, and vanillin, which is a common flavorant in perfume. So the possibilities really don't stop there. Biological synthesis gets crazy. These are two papers that have frankly inspired me in my route to graduate school. This is really what gets me excited. So if you think I'm weird for that, that's okay. But I think this stuff is crazy. On the left side, you see an engineered strain of E. coli that can directly degrade PET, which is the plastic that makes up plastic bottles, and turn it into vanilla. So the people who made this paper took plastic off the street and stuck it in a little bath with these E. coli, and were able to make something useful from a feedstock that's not chemically accessible, and honestly something that we're really having trouble getting rid of. On the right side, you see this paper where the authors studied the pathway for the biosynthesis of cocaine in coca leaves. So this is something we didn't really know exactly how it worked before, but the authors were able to put together this complex pathway. And not only did they do that, but they put it back together in tobacco leaves. And so they were able to reconstitute a pathway to make this complicated molecule, which, yes, it's cocaine, but it's not just any molecule, it's a highly bioactive molecule, you know that. And so you know that now we can not only take plastics, but we can also take carbon dioxide and turn it directly into these complicated molecules that might have medicinal properties. So a class of proteins that can accomplish a lot of really interesting biochemistry are heme proteins. So naturally, you know of hemoglobin, which binds oxygen in your blood and carries it, makes it soluble. But there are a lot of other proteins that accomplish synthetic tasks. Heme is sort of like this big aromatic molecule that binds a metal in the center. This allows it to accomplish a lot of tasks for redox chemistry. It can also stabilize these high energy intermediates like carbines and nitrines to allow for, with a bit of engineering, some really weird reactions. So I'm showing you from the lab of Francis Arnold, where engineering around the active site of a heme protein allows for the formation of carbon-silicon bonds in biology with remarkable stereoselectivity. And another interesting reaction, which is technically available in biology, but where this compound here was coupled to an alkene to make both isomers of a cyclopropane. And so what I'm trying to show you here is not necessarily all of the functions of heme, but just the idea that these heme proteins can allow us to do really niche chemistry in a green way. A protein that I'm really interested in engineering is this one, Cytochrome C. So it's a small heme-containing protein with a little bit of a nuance from some of the more commonly known heme-containing proteins. Now, in hemoglobin, the heme group sits in a little pocket very comfortably, but it's not stuck there. It's sort of just hanging out, chilling in a fold on the protein surface. But in cytochrome C, the heme group is actually covalently attached through this peptide motif, cysteine, 2 spaces, and then another cysteine and a histidine. Whenever the heme is brought in the presence of this motif, as well as another enzyme catalyst, it is covalently connected. Now, this confers a lot of properties to the heme that might not otherwise arise just from non-covalent attachment of heme to a protein surface. So it really gives us a lot of play as far as engineering the properties of the protein for catalytic function. Just want to show you a little bit more of the difference between covalent and non-covalent hemes. On the left side here, I have deoxyhemoglobin, and on the right, I have cytochrome C. So what I'm hoping to illustrate here is this idea that heme chemistry is very sensitive to environment. So there are a lot of parameters in the active site of a protein that one can change to get new function out of the protein. So the first one is the ligand or coordination sphere around the metal center of the heme. So just to describe what that means really quick, you can see a heme right here, and this orange, it's a little hidden, but the orange there is the iron atom in the center of the heme. These blue groups surrounding are histidines, which are nitrogen-containing amino acids, which are able to bind to the center of the heme. In hemoglobin, only this one is actually bound. The other one's sort of just hanging out, ready to bind oxygen. But in cytochrome C, there is a histidine bound as well as a methionine. And so by binding nitrogen and sulfur, we get some different chemistry. Here I'm showing the maximum absorbances for both proteins, which is just kind of an analog for a couple other properties of the electronic structure of the heme. Not only can you change the ligands of the heme group, but also the shape. You can see here that although normally you would expect a large aromatic compound to be fully planar, the heme can sort of fold in on itself in multiple different ways, and this is caused really by some almost mechanical force that's exerted by the protein, and it's much more dramatic whenever we have heme attachments. This is another reason why we're interested in pursuing covalent hemes. Finally, substitution of the metal center will always greatly affect the chemistry of a heme-containing protein. You can see here an example of a truncated version of cytochrome C, which, whenever substituted with cobalt, became a hydrogen evolution catalyst in neutral water. So we're really interested in working more in exploring these parameters. Clearly, we can change the chemistry of heme-containing proteins a lot by changing the ligands, the shape of the heme, or the metal center. So why has more engineering not been done in this field? Really seems like it's promising, right? The answer to that is because the biosynthesis of cytochrome C is very, very complicated. There are multiple systems to biosynthesize covalent heme proteins, but I'm only going to show you here two. So the one on the top is hollow cytochrome C synthase, or HCCS. Now, this is a small soluble protein, so it floats around in the cytoplasm of the cell, and it's highly specific to cytochrome C. So it can only recognize that substrate, and in the presence of that substrate in heme, it will go through this process of binding a heme and binding the protein, and then forming a covalent attachment and releasing it. It doesn't recognize any other substrates with this CXXCH heme attachment motif. On the other hand, cytochrome C maturation system 1, or CCM1, is a membrane-bound complex with many enzymes, 10, that is able to attach heme a little more loosely. It's able to recognize maybe some other proteins with heme attachment motifs. Now, because this is membrane-bound and all of the proteins are actually sort of facing in towards this little space in between the two membranes of E. coli, it's not as easy to engineer. There are 10 proteins in this group, so where do you start if you want to change the biosynthetic function? We really prefer to be changing one protein and not 10. So that's great, and we now have this defining objective that we'd really like to encourage HCCS to behave more like CCM1, to relax a little bit, recognize some other people, and do some other work, do some more biosynthesis. But in the process, we'd also like to pull in other transition metals because, like I said, this is really the most dramatic way to change the function of a protein that contains a metal. Completely change the metal and you'll get new chemistry. So it sounds simple, but the problem is that we really only have a vague idea of how HCCS works. We don't have a structure of the protein to look at, and there have only been a couple biophysical studies of the mechanism. And so it's not so easy to just ask it to do this. Enter directed evolution. So this is a really, really cool class of experiments where we can essentially tie the function that we want. So in this case, I'm calling it HCCS star activity, or the incorporation of a metal substituted heme to a growth advantage. And so this means that whenever a synthase variant does what I want it to, the cells will be able to live. They'll survive in the presence of a toxic compound. And on the other hand, we can tie undesirable wild type activity, or the incorporation of iron substituted heme, to a growth disadvantage. Cells that are continuing to express proteins with the activity that I don't want will no longer be able to grow. So in order to do this, we proposed this system. So it's a lot here, but I'll just run through really quickly the 3 DNA vector system that we proposed for directed evolution. There are a lot of components that go into a directed evolution system. The most important, perhaps, is the selection pressure itself. So we proposed PHSDS 2.2 to accomplish this goal. This vector encodes for 2 proteins, which are mutants with heme attachment motifs to enable selection. I'm just going to explain briefly how that works now, and we'll go into more detail later. But the 2 substrates that we chose are first uracil phosphoribosyl transferase, or UPRT. This is an enzyme that, in the presence of 5-fluorouracil, will produce a very toxic derivative, 5-FUMP, which will arrest cell growth. We hypothesized, though, that if we could convince HCCS to attach heme to UPRT, we could actually shut down its function. And so cells that are able to do that would be able to survive in the presence of this otherwise highly toxic compound. On the other hand, we have chloramphenicol acetyltransferase, or CAT, which is an antibiotic resistance gene which makes bacteria resistant to the antibiotic chloramphenicol. Sort of to the converse, we hypothesized that attachment of heme to this would cause cell death. Cells would become sensitive again to chloramphenicol. So by tuning the conditions of growth and alternating between positive and negative selection, we think that we might be able to just select for this HCCS star activity and not HCCS. Now that's contingent on being able to grow cells in the presence of metal substituted hemes. We show here that we want them to add this extra supplied heme, which is in blue. But hemes are, like I showed you earlier, these really big flat molecules with a lot of hydrophobic surface area and a little bit of hydrophilic surface area. And so that is something that looks very perfect to get incorporated into a cell membrane. So you can't just sort of give a cell a heme and ask it to eat it up. It's not going to do that. On the other hand, there are, fortunately for us, some, well, only in this case, fortunately for us, some bacteria which natively like to get their iron from your blood. And so there are actually systems that allow for bacteria to pull heme directly out of the environment around them. And this is PHUG21. So this vector expresses a little sort of sextet of proteins that allow us to take up heme from the surroundings. And finally, if we're going to test mutant varieties of HCCS, we need to make mutant varieties of HCCS. And so that's this library down at the bottom. So this is the system, but the first 2, the first and third, PHSDS 2.2 and POKD4 HCCS, are things that we sort of made up to do this, and this turned out to be a very cumbersome system. And so there are a lot of questions of how do you actually put this sort of thing together? How do you create and combine all of these DNA vectors to encode and express proteins for a directed evolution experiment? So I'd like to start by explaining how we created PHSDS. And this is our platform for selection. So I told you earlier that the goal was to come up with a way to turn off the activity of these proteins for using heme attachment. So it may not be obvious how that works. So I just want to tell you a little bit about how the protein UPRT accomplishes its function in order to maybe give you a better idea. So that's this figure right here. UPRT attaches a nucleobase to a sugar backbone. And in the process, there's a dissociation of a phosphate group that forms this transient oxocarbenium intermediate. So in protein chemistry, something that we really like to think about is substrate sequestration, the ability of an enzyme active site to wrap around a really reactive intermediate and hide it away from water. So this is something that would really not want to form in water, or at least not in any appreciable manner. But in a protein active site, even though it's high energy, it's still stable. On the other hand, we thought, well, that requires some flexibility in the active site of the protein. So what if we add a heme group to that? Could we actually lock up the active site and keep water in there so that this is no longer able to form, UPRT can no longer accomplish its function, and in the case of that protein, cells are given a growth advantage. So we used a similar rationale for CAT and looked for sites near the active sites of both proteins with solvent-exposed residues to incorporate heme attachment motifs. So for UPRT, I only chose one, but for CAT, I actually chose a more exposed residue that was a little bit further away from the active site, and one that was directly involved with the active site, but more secluded. So there are a couple reasons for this, but I guess the quick and dirty explanation for why we chose two is just for more tunability. If we could incorporate two heme attachment motifs into CAT, then we have a wider variety of sort of strong heme attachment or maybe easier heme attachment that we could access. So we proposed this system, and now we want to put it together. This starts with the polymerase chain reaction. So something that I wanted to tell you earlier, but I'll tell you now, I guess, is that it's really difficult to synthetically change the structure or the sequence of a protein. It's really not easy to access in such a large polymer of maybe 100 amino acids, maybe 200, maybe 500, just a specific residue or 3 specific residues and change them to be what you want. On the other hand, in biochemistry, we really like to use this DNA molecule, this long-term storage of information, in order to quickly change the sequence of a protein, because DNA encodes for RNA, and RNA, these little short messages, allow us to translate proteins. We can just change the sequence of DNA and then get new proteins that way. And so, using the polymerase chain reaction, we're able to accomplish exponential amplification from a small target, a specific fragment of DNA that we want. This is using a polymerase, which is just a really smart copy enzyme that can look at the sequence of DNA and reproduce it. And these short strands of DNA called primers that allow us to mark specific sites on a target for amplification. So after a lot of temperature cycling, we get to the specific copies that we're looking for. And you can see here the yields are very good because this is exponential amplification. Starting from a single copy allows us to make 10 billion copies in 30 cycles. This is a really easy method to copy DNA and edit it slightly to get a new fragment that you're looking for. Now, whenever you have these fragments, you need to put them back together because the DNA that we actually want to put into a cell is typically in the form of a plasmid or a vector, which is a circular piece of DNA. It's more stable, and it's typically the way that DNA is stored in cells, in a circular form. And so once we amplify the fragments, CMR is the gene that encodes CAT, chloramphenicol acetotransferase. SCMR is just a tag for another antibiotic resistance gene that we use to select for this plasmid. UPRT is the gene that encodes for UPRT. We put them together in a little bath and used Gibson assembly, which is just a little cocktail of enzymes that looks for overlaps and stitches together fragments. And we had the plasmid for heme switch dual selection, or PHSDS. What I'm showing you here is just a gel. This is the first molecular cloning experiment I ever did, and so I was really excited whenever I saw that it worked pretty well. This is exactly what we would expect to see for this plasmid. So moving forward, I put together those 3 genes, but I haven't actually incorporated the heme attachment motifs yet. So the whole point of this is to add a CAT and UPRT gene with heme attachment motifs. The way that we accomplish that is through site-directed mutagenesis, which is just a little derivative of PCR amplification, where we edit our primers to contain the sequence that we want. And so these primers are still able to mark and target specific sites on proteins, or on DNA, but there's a slight change in the code of the primer that eventually leads to a very high yield of a new sequence rather than the original wild type sequence. And Once we had those fragments, we again stuck them into DNA, grew them up, and sent them up to Cornell for Sanger sequencing, which is yet another kind of derivative of PCR. It's linear amplification instead of exponential, but it uses fluorescent tags to help us figure out the sequence of DNA. So once we set it up and realized that the sequences were exactly what we wanted them to be, we were able to move on with PHSDS. Now, over time, nature spends a lot of energy and work evolving proteins to do exactly what it wants them to do. And so whenever you incorporate mutations into a protein, it's reasonable to expect that you took what nature thought was best and made it something different. Probably not going to be as good as it used to be anymore. And so the purpose of the experiments that I'm going to show you now are to see really how did we affect the proteins that we mutated. We've incorporated these motifs at the active site, probably the most important part of the protein. Are they still going to be active? And to check for this, all I did was I grew my mutants in the presence of a gradient of chloramphenicol or 5-fluorouracil, so just the substrates of the respective proteins. And I put them in a plate reader. So this was really cool. I was able to essentially, over the course of a day, collect data points every couple minutes and generate these growth curves. So this is literally just how dense each culture was over time. And it was really awesome to see that this worked about exactly as I would expect it to work. Putting all of the data together, I was able to collect these tables. On the left side, I'm showing you the UPRT mutants. So you can see that as we increase the concentration of 5-fluorouracil, they're making more toxic metabolite, and so growth rates are going down across the board. This PRED Cas9 is just a control, which doesn't express for UPRT. So you see at low concentrations, there's really no effect. As the concentration increases, there's still some interference, which is perhaps to be expected for something that becomes toxic. But comparing the wild type with PHSDS 1.2, we see that there's essentially no loss of activity. A very slight increase in growth rates, as you would expect for a loss in the activity of a toxic gene, but it was tolerated, which was really exciting. On the other hand, for cat, you can see a little bit of change. So comparing the wild type again, PHSDS 1.2, with 2.2, which was the solvent-exposed site, we saw pretty comparable activity up until high concentrations where there was a little bit of a drop-off. Now I will say that this is an extraordinarily high concentration of chloramphenicol, and more than you'd ever actually use, but you can still see that there is a difference in activity, which was, again, expected. On the other hand, for the solvent-sequestered site, we saw a complete loss of function. And it's actually pretty obvious why this happened if you think about it. What we did was we mutated residues which were very hydrophobic and sitting in the core of the protein, and we switched them out with something that had a different shape. And while cysteine is sort of hydrophobic, it does sometimes like to sit on the inside of a protein and not in the presence of water. It still is a different shape than the amino acid that we kicked out. And so this is disrupting the fold of the protein, and that's likely the reason why we lost the structure and therefore the activity. On the other hand, it was really nice just to see that these experiments showed activity for at least one of the cat mutants and one of the UPRT mutants, because this means that we have active variants of both proteins with heme attachment motifs near the active site. So that is PHSDS finished and ready to go for directed evolution. But if we want to do directed evolution in the presence of metal substituted hemes, we need metal substituted hemes. And this was an interesting one for us because I'm showing you a synthetic scheme here. This is protoporphyrin 9. It's very cheap. Metal substituted porphyrins are not cheap. This is probably 5% to 10% the cost of this. And so rather than spending $2,000 on substrates, we thought it would be a lot nicer if we could synthesize them. And as it turns out, the synthesis is incredibly easy. You could probably do this in Gen Chem if you wanted to, except for it's really expensive. So you really just put in the protoporphyrin 9 with whatever metal you want to sit in the center, heat it up, look for fluorescence to go away, and when it's gone, you just dump in water and filter it. It does not get easier than that. I say that the yields are greater than 90%, but I would hazard a guess that they're probably greater than 99%, and all of the loss is from the stickiness of the protoporphyrin to the filter. So this is a really, really easy reaction. And it was nice to be able to generate 5 transition metal hemes, all of the hemes from manganese to zinc, except for iron because it's cheap and we just buy it, in like a week. So once we got those, we of course wanted to make sure that all 6 of the hemes that we had were exactly what we thought they were. So to characterize them, we used UV visible spectroscopy. This is just looking at the absorbance of the compounds in the presence of UV and visible light. And I just want to remind you, one of the key parts of this sort of experiment, I told you earlier that heme chemistry is incredibly sensitive to its environment. And this is no exception. Whenever you put a heme in solution, if the solvent is a Lewis base or if it's polar, it's going to want to stick around that metal center, and it will often covalently bond to the center in some way. And so it's really not practical to just look at the spectrum of a metal substituted heme in just solvent. On the other hand, there's high affinity for nitrogen-containing ligands to the metal center. So just spiking it with something like piperidine or maybe imidazole or pyridine allows us to get very reproducible spectra. So I don't want to spend too much time here because this isn't the key, but I do want to show you just this moving spectrum. So we have a standard, which is the unsubstituted porphyrin, and then a porphyrin with no ligand and a porphyrin with ligand. And you can see that as we go along, as I just said, it moves towards the red wavelength. So just one more thing here, I have this table of all of the key absorbances of all of the porphyrins, and you can see that for all 6 of the hemes, there are characteristic spectra, which is great because none of them are the same, which means again, we have what we thought we did and not something else. So now that we have metal substituted hemes, we just need a way to get them into the cells. And I told you about PHUG21 earlier. It's this big vector. It's about 5 times the size of PHSDS 2.2, the vector I made earlier. And that is inconvenient. DNA is pretty fussy whenever it's this large. It's difficult to get into cells. But we needed it because it expresses this very useful system that allows for hemes to be pulled through this active transport protein into the periplasm, just a little space between the 2 membranes of E. coli, and shuttled into the cytoplasm. So it was challenging to get this into E. coli. It took me about 4 months of work to even extract the vector out of bacteria, which is normally a very easy process. It's something for any other vector I can do in a day. But this took about 4 months. But finally, after so much work, I was able to get it out. And we did an experiment called a restriction enzyme digest, which is just putting in a very specific enzyme chainsaw to cut at these sites and produce the fragments that we expect. And so after that work, we were able to confirm that indeed we do have PHUG21. So now we have these 2 parts. We have a selection vector and we have heme uptake. So the last ingredient that we need is a library of variants. So in comes the design of the HCCS library. I mentioned earlier that we don't know exactly how HCCS works to attach heme to cytochrome C. We have a lot of ideas, and there are a lot of experiments mutating just little parts of the enzyme and seeing, how the products come out. But we don't really know physically how heme binds to the protein, how it catalyzes the addition of heme to cytochrome C, and how it releases. But the other issue is that there's no actual experimental structure of this protein in the free market, as in PDB, the protein data bank. And so we needed to come up with a solution. Fortunately, there's this awesome software called AlphaFold, which is a machine learning program that can de novo or from the ground up simulate the structure of a protein. So that's if you don't know that that's crazy, that's crazy. That we don't have to generate a protein structure using crystallography, a really difficult technique. You can just simulate the structure of a protein, and it's pretty reliable. And so using that, as alongside the study I mentioned earlier with just little mutations, I was able to explore the active site. And I landed on mutating these 3 residues to generate my library. So I'm showing in blue here this histidine, which is histidine 133. That is the ligand that binds heme and sets it up for attachment to cytochrome C. But there are these other residues. So tryptophan 97 is in a really good position if heme is attached here to stack on top of it and through aromatic interactions or pi system interactions, stabilize the addition of that heme. And then we also have N137 or asparagine 137, which is in the right position to either donate or accept a hydrogen bond from the imidazole. That's a really interesting one for us and one that we're curious to see where it goes, because modulating the basicity or changing the essentially bond strength of this imidazole to a metal center could really have a lot of advantages for us. finally, we chose glutamate 138, and the function of that residue in the addition of HCCS and heme is unknown, but we know that it impacts heme binding according to the paper that I mentioned earlier. So with these three residues together, we generated a library through randomization at those three positions, we call it site saturation, and introduced additional genetic variety using error-prone PCR. The reason we chose to do this is because, as I said earlier, HCCS is highly specific. It only wants to play with Cytochrome C. It doesn't want to add heme to anything else. And so by adding additional mutations to holocytochrome C synthase, we're hoping that we can allow it to relax a little bit and recognize other substrates. So error-prone PCR is essentially just taking a less smart polymerase, or one that isn't able to proofread itself, and giving it a concentration of some metal ions, and it's able to introduce some really useful random point mutations for us. Again, I'm showing you here this gel, which is 14 different variants of the library that I extracted and sent off for sequencing. And that showed us that, yes, the mutations worked. The randomizations worked very well, and the point mutations were throughout the gene. So we have all the pieces together, finally, and we're able to combine them in cells. In order to do the actual directed evolution experiment, we need to put all of these proteins together. So we need to put all of the vectors in one cell line and allow it to express the proteins and grow in the presence of different substrates. So to start that off, I combined PHSDS 2.2 and PF21 into the BL21 strain of E. coli, and moved on to transform the POKD4 HCCS library to even positive and negative selection. Last thing I want to tell you about before I tell you about the directed evolution experiments is how we actually get DNA into cells. So I chose to wait until now, even though I've referenced putting vectors into cells a lot of times, because it's really most relevant in this case. Whenever you make a library of mutants, the idea is that you want to look at the function of all of them. You want to see how all, in this case, 8,000 times n mutants work. So we did a site saturation library with 20 different amino acids at 3 positions, that's 8,000. But once we did error-prone PCR, every single molecule of HCCS that gets produced by every single variant of E. coli that takes up a plasmid will be different. That is an infinitely large library, or as large as we can transform. And so we really want to have a high efficiency. There are multiple methods to be used for for transformation. The most common is probably chemical transformation, which is a very cheap method where a quick application of heat polarizes the cell membrane and pops open little holes for DNA to enter. It's convenient, but it is not efficient. And so whenever we want to achieve maximum efficiency, we look to electroporation. It's a much more transient, quick pulse of electricity that pokes similar holes in the cell membrane, but results in a greater uptake of DNA and a higher rate of survival. That allows us to get higher transformation efficiency. So again, with all of this together, I was able to, in parallel, screen 12,000 variants of HCCS, or 24,000 between 2 experiments in the presence of nickel and cobalt heme. It took a long time. I flipped them between conditions of positive selection in the presence of 5-fluorouracil and metal substituted heme, or negative selection in the presence of chloramphenicol and the natively synthesized iron heme. And after 14 days, I was really excited to see, like how many variants of HCCS did we pull out? Did we get anything that really worked? I was super excited. And so I came up with this idea of this really fast, high throughput method to generate samples for screening. It's called colony PCR. I didn't come up with this, but I sort of developed the method for the lab, where you scrape off a little bit of bacteria from a plate and you put it in a PCR tube. Normally you want to purify DNA to use as a target, but it would be really nice if we could just skip all of that time and put it right into a tube. It turns out you absolutely can. You just stick it in a tube, and you nuke the tube for a long time at a high temperature, and it pops the cells right open, and all the DNA you would ever want is right there. And so we were able to use this method to amplify out this control fragment, which is exactly at the height we would expect for the HCCS gene. On the other hand, after screening like 400 different, it's probably not 400, but it's probably close to 200 different cell lines, looking for the reason why this wouldn't work, I realized that the strongest band in all of the experimental assays that I did corresponded to the vector with no insert. And I realized that our directed evolution experiments had kicked out the gene entirely. So this was frustrating, and I really wasn't sure why it worked, or why it happened at first. And then I realized that it was actually kind of a good thing in some ways. There's a saying in directed evolution that you get what you select for. And in this case, I think that we probably just got what we selected for. There are a lot of causes, but the main one really is just that we may not have given it the right selection conditions to draw out the mutants that we're looking for. We may not have put enough positive selection pressure on the cell lines, meaning that even after a little bit of benefit from HCCS adding metal substitute heme, maybe they didn't grow any better. Or maybe negative selection wasn't working either. On the other hand, it's possible that our growth benefits were not very useful, and so overexpression of synthase would be a real detriment to the cells. Expression of protein takes a lot of effort for a cell, and so if you only get a 1% growth advantage, but you lose 5% of growth rate from protein expression, you'd rather just not make any protein at all. And lastly, something that I only realized after, we needed to switch backbones to carry the library into the triple transformant. But I realized that the cloning experiment may have actually been diluted with failed clones, and so it may have included, from the get-go, many different vector backbones with no gene at all. So these would have a great advantage, of course, if, again, the selection conditions don't help at all, then not expressing protein is really great for those. Fortunately, we know a lot of the route forward. There are a lot of ways that we can overcome these challenges. We can work on tuning the expression levels of HCCS or changing the concentrations of the selection pressure for negative and positive selection. We can improve the recognition of the mutant substrates by Cytochrome C synthase by taking advantage of a secondary recognition sequence found on the original substrate. And we can optimize the library assembly and delivery. So get rid of these diluted, failed clones that we're not looking to screen for, and try to increase the transformation efficiency. So maybe instead of screening 12,000 clones, we could be screening 100,000 clones or 1,000,000 clones, or even more. So that's where this story ends for me. But I'm really, really excited with all of the prospects for how this project can move forward to see where it goes in the future. Do have some time for questions, but before we get there, I do want to say some thanks and acknowledge the people who made this project possible. Honestly, looking around, all of you in some part have had just an immense impact on my ability to do this work and stay sane. This isn't easy, so probably every single one of you has solicited a conversation from me, you know, being frustrated, being ready to quit, being ready to walk away and just do something else. I don't know, pick up music. But I didn't, and I'm really proud that I didn't, and I'm really glad that I'm still here and here today to tell you about this. I want to thank all of my lab mates who have specifically spent time in the lab with me and have collaborated with me, talked with me about this project and things to change. I want to name Sarah Matier, who has spent countless hours in the lab with me, who has talked with me about this all the time, and most importantly has really had her hands in this project as well. I couldn't have finished this without all of the work that she's done collaborating with me on both experiments, preparatory work, and all of the like. I want to thank all of the faculty of the Department of Chem and Biochem. I've talked with you in person about how much impact you've had on my life, but everybody else should know too that I would not be the person that I am today, and I wouldn't be here doing this, moving on to do graduate school if it weren't for all of you. And so I'm immensely grateful. And most importantly, I want to thank my mentor, Jesse Kleingardner, who has had an indescribable impact on me, both as a person and as a scientist. I don't want to cry, so I can't talk anymore, unfortunately. Yeah, there's too much to say, so thank you. I thank you all for your time. This has been a really, really awesome opportunity to tell you about my research work, and I would love to take any questions. Thank you.
Questions?
So were you saying at the end here, the gene wasn't uptaken at all? So you're saying if it had worked, if the transformation worked, then the selection process would have worked. But if it didn't have it in the first place, it wouldn't have mattered?
So it's impossible to say. Really, the reason that we made such a large library is because we don't know what's going to work. We don't know exactly what mutations are going to do what we want them to. And so the goal is to introduce as many different variants as possible. I didn't have the time to figure out exactly how efficient the cloning was. And the cloning efficiency just means how many successful variants did we make out of how many failed variants. And so it's hard to say, but I would say probably a mixture of both. There were many failed clones, and even if it was only 10% failed clones, maybe the 90% just weren't what we were looking for, and that's okay. This often takes a lot of rounds and a lot of trials.
Just curious, why did you transform into BL21 cells and not T7 Express?
So we chose to do BL21 mostly because the looser translation, so for everybody else, basically BL21 is like, it leaks out protein, and so even if you don't tell it to express protein using a chemical messenger, it will still make some, whereas T7 Express is really tight. It only makes protein if you tell it to. And so because we don't really know how much of the protein, the synthase that we want to make, we thought it'd be better to just stick with old reliable and not do the new variant that's maybe gonna give us more trouble or do something we don't know. It's more studied, more used. Yeah.
During the 4 months of failure, what did you do that kind of jump-started the success?
Well, it's a funny story. So I emailed the person who sent it to us, and he was like, "Oh, well, we actually had some like compromise. You know, there's a whole story. It was really funny." And he sent us more. And when he sent us more, he also told us that, and I did a lot of reading in literature, basically, of just how to get DNA off of the purification membrane. So whenever you do it, there's like a little polar membrane that it sticks to, and then you push it off. It basically just took hot water. That was the only thing. I had to heat up the water, and then it came off. So what did I do in the meantime? I just like tried it over and 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 over again, and it didn't work. And then I heated up the water, and then it worked perfectly. So that was like a little annoying, but I had what I wanted. So in the end, just move forward.
Other questions? Okay, well, let's thank Noah again. He'll be going to the Chemistry Biology Interface program at Johns Hopkins University next year. I'm going to ask you all, except for faculty members in the department, to make your way out.